primary nucleotide sequence database. Nucleotide sequence databases organize and distribute nucleotide sequences from all available sources. They contain sequences submitted directly by scientists and genome sequencing group and sequences taken from literature and patents. The International Nucleotide Sequence Database consists of the three primary nucleotide sequence databases. 1. GenBank, that is National Center for Biotechnology Information. Number 2, EMBL Nucleotide Sequence Database or European Molecular Biology Laboratory. Number 3, DDBJ or DNA Data Bank of Japan. GenBank is physically located in the USA and is maintained by National Center for Biotechnology Information and accessible through NCBI portal over internet. EMBL or European Molecular Biology Laboratory is in UK and DDJB or DNA Data Bank of Japan is in Japan. The three primary databases are repositories for nucleotide sequence data from all organisms. They accept nucleotide sequence submissions and then exchange new and updated data on a daily basis to achieve optimal synchronization between them. Historically, the three started out as separate databases with independent means of collecting the data and with different formats for arranging it. Now, the three together form the International Nucleotide Sequence Database collaboration. They have uniform format and exchange data on a daily basis with the result that it is sufficient to connect to any one of them in order to access the sequences present in all the three. The multidisciplinary research group consists of scientists from diverse fields who work on the management of biological data as well as to develop bioinformatic tools to analyze biological data. Data can be exchanged between the three collaborating organizations on a daily basis. The nucleotide databases have reached such large sizes that they are available in subdivisions that allow searches or download that are more limited and hence less time consuming. For example, GenBank has currently 17 divisions. There are no legal restrictions on the use of the data in these databases. However, there are patented sequences in the databases. Having your sequence in the database means it is readily available to the scientific user community. A repository of primary nucleotide sequence data that is freely accessible is essential for computational analysis and genome research. DNA sequence information can be obtained at the following three levels: genomic sequence level, which include information at the level of untranslated sequence, exons, introns, mRNA and cDNA, that is transcription level, protein, that is translational level. Thus, DNA databases contain a variety of different types of data. The genomic sequence data can be further divided into three categories: full length CDs, partial sequences including EST and STS, genomic sequences including HTG and GSS, all of the rest sequences which include SNPs. The ESTs and other partial sequence data such as sequence tagged sites or STS are usually short sequences, whereas full length sequences making up the rest of the database tend to be much longer. The description of precise location, size and function of sequence elements within a DNA, RNA or protein and the prediction of function from sequence refers to annotation. A genome must be annotated to provide the putative start, stop and structure of each gene along with predicted gene products and about similarities to other known or predicted genes and proteins. The Genomic Single Sequence Database or GSS contains single pass genomic sequences, exon trapped genomic sequences and ALUI PCR sequences. HTG or High Throughput Genomic Sequence Database contains unfinished DNA sequences. NCBI. The National Center for Biotechnology Information (NCBI) was established in November 4, 1988 as a part of the United States National Library of Medicine, a branch of the National Institutes of Health. The NCBI is located in Bethesda, Maryland. The NCBI houses genome sequencing data in GenBank and an index of biomedical research articles in PubMed Central and PubMed, as well as other information relevant to biotechnology. All these databases are available online through the Entrus search engine. NCBI is directed by David Lippman, one of the original authors of the BLAST sequence alignment program and a widely respected figure in bioinformatics. What does NCBI do? NCBI creates public databases, conducts research program in computational biology, develops software tools for analyzing genome data, and disseminates biomedical information, all for the better understanding of molecular process affecting human health and disease. NCBI develops biomedical database, promotes standards for databases, data deposition and exchange. To help the scientific and medical communities, they develop, distribute, support and coordinate access to a variety of databases and software. They also conduct research on fundamental biomedical problems at the molecular level using mathematics, statistics and computational methods. In October 1992, the NCBI, the largest biomedical research facility in the world, took liability for the GenBank. In addition to GenBank, NCBI also supports and distributes a variety of databases for the medical and scientific communities. These include the online Mendelian Inheritance in Man or OMIM, the Molecular Modeling Database MMDB of 3D protein structures, the Unique Human Gene Sequence Collection UniGene, a gene map of the human genome, the Taxonomy browser and the Cancer Genome Anatomy Project or CGAP in collaboration with the National Cancer Institute. GenBank. GenBank was set up in 1979 at the LANL, Los Alamos. Since 1992, GenBank is maintained by the NCBI Bethesda, USA. It is the most comprehensive and annotated collection of DNA sequences and is a part of International Nucleotide Sequence Database Collaboration INSDC, which consists of DDBJ, EMBL and NCBI. Out of the 3 nucleotide sequence databases, GenBank is the major contributor of sequence data. A new release of GenBank is made every 2 months. Sequence information are incorporated from direct author submissions, large sequencing projects, the Genome Sequence Database, Santa Fe, USA, the United States Patent and Trademark Office, USTPO, and other international patent offices, and via exchange from partners of the INSD. Direct submissions to GenBank are prepared using one of 2 submission tools, Bankit or Sequin. Information can be retrieved from GenBank using NCBI's Entrus retrieval system. GenBank may also be searched with query sequences using NCBI's web interface to the BLAST suite of programs. Each entry consists of a number of keywords, associated sub-keywords and an optional feature table. How to retrieve information from NCBI? The web link to NCBI is www.ncbi.nlm.nih.gov. From the drop down, select protein to retrieve protein sequence data. Then select nucleotide to retrieve the nucleotide sequence data. Finally, type the protein name or the accession number and click search. Next, select any one of the entry. If the query is the NCBI accession ID, the details of that particular ID will be displayed directly as shown here. Each entry consists of a number of keywords, associated sub-keywords and an optional feature table. The keywords include definition, a concise description of the sequence, for example, lipase from Pseudomonas fluorescens. Accession. Accession number is a unique constant code assigned to each entry, for example, M13667. Version. This indicates the version of any sequence revisions. Keywords. This introduces a list of short phrases assigned by the author describing gene products and other relevant information about the entry. Source. It provides information on the source from which the data has been derived. Sub-keyword organism illustrates the biological classification of the source organism. Reference. This indicates the position of the sequence data to which the cited literature refers. Sub-keywords include authors, title, journal, medline and PubMed pointers, etc. Comment. A free text section that allows any additional annotations to be added to the entry. Features. Marks the feature table whose purpose is to describe the properties of the sequence in detail, such as the gene name, coordinates of the coding sequence. Database cross-references made through the /db_xref qualifier and to its sequence translation in GenBank's peptide database, for example, GI190470. Base count details the frequency of the different base types in sequence. Origin. This notes the location of the first base of the sequence in the genome. Then the nucleotide sequence itself follows and the entry terminates with ##. To access to GenBank as to all databases at NCBI is through the Entrust search program. This front-end search interface allows a great variety of search options. More importantly, it enables searching more than one database at the same time, interpreting the search parameters in an appropriate way for each database. Here you can see the home page of Entrust search engine. Recently, the NCBI Entrust search engine has been modified as gQuery, NCBI Global Cross Database Search. Entrust is NCBI's primary text search and retrieval system that includes DNA and protein sequence, structure, gene, genome, genetic variation and gene expression. It includes about 40 molecular and literature database, and these linked databases are listed as shown here. Biosystems, books, conserved domain, DBGAP, DBWAR, epigenomics, EST, gene, genome, genome project, GEO datasets, GEO profiles, GSS, homologene, MESH, NCBI website search, NLM catalog, nucleotide, OMIA, OMIM, PopSet, probe, protein, protein clusters, PubChem bioassay, PubChem compound, PubChem substance, PubMed, PubMed Central, SNP, SRA, structure, taxonomy, UniGene and UniSTS. To submit sequences in a database, submission tools are available. BankIt is a worldwide web-based sequence submission tool which can submit sequence directly to GenBank. Another tool, Sequin, developed by NCBI, is standalone submission software. Sequin can also be used to submit and update sequences in DDBJ and EMBL. A user can submit single short sequence, long and complex submissions, segmented sets of DNA, phylogenetic, environmental, mutation, population studies and multiple annotations with Sequin.